Hi, everyone. So my name is Felicia, and I am a project coordinator at Cleveland Water Alliance. And I'm so excited to welcome you all to the Detroit quarterfinals, um, hosted by TechTown Detroit. This is our very first quarterfinal, but be sure to tune in to Cleveland's this upcoming Thursday. Before I transition to Paul Reiser from TechTown to give his opening remarks, I just want to thank our sponsors, partners, and supporters. Erie Hack is presented by the First Solar Community Giving Fund of the Greater Toledo Community Foundation and the Northeast Ohio Regional Sewer District. Erie Hack is supported by OHM Advisors in partnership with Midstory, the Blackstone LaunchPad at the University at Buffalo, WEtech Alliance, and of course, TechTown Detroit. With that being said, I will now let Paul take over from here to introduce the judges and to provide his opening remarks.
Thank you so much, Felicia. Good afternoon, everyone. The time has finally arrived, and it's awesome to be here virtually with each and every one of you. As Felicia mentioned, my name is Paul Reiser with TechTown Detroit, proud host of the quarterfinals for Erie Hack for our region. Would like to formally welcome everyone to this Tuesday, October 19th, 2021 Detroit Erie Hack quarterfinals. And this is Erie Hack 3.0. So we've had a great series of past Erie Hacks in past years. For those of you who are familiar with or who have participated in some way with past Erie Hack events, welcome back. And for those of you who are new to the program, a big warm welcome as well, and thank you for joining us. We're in for a great program today and lots of fun. So I have the great pleasure of having been involved with Erie Hack since its inception year, which was in 2017. Please be reminded that our regional semifinals are November 4th, and our finals will be held November 18th in hybrid fashion. So I would like to ask each and every one of you to tell your friends, colleagues, coworkers, your neighbors to tune in and get connected with the Erie Hack movement. As I like to emphasize and remind anyone that I can in every opportunity, we all have a role to play in ensuring that our most precious asset is not only protected, but prioritized as a resource that we all care for and provide the utmost respect for. And a lot of that has to deal with amazing innovation that some of you are bringing forth today and that others will be able to witness. Additionally, I want to make sure that I send a huge thanks and words of appreciation to the Herb Family Foundation for their tremendous support in Southeastern Michigan and across the Great Lakes for many years now. We absolutely could not do this without you. So again, immense thanks and appreciation to the Herb Family Foundation, our key sponsor and supporter in the Southeastern Michigan and Southwestern Ontario Erie Hack delivery, as well as their support is deeply appreciated across all of the Erie Hack participating cities. Erie Hack is made possible by many great partners, supporters, innovators, coaches, mentors, and ecosystem builders. So thanks to each and every one of you as well. I'd be remiss to not give a huge thanks as well to the Erie Hack team members, Felicia, Max, Brian, and the entire team. We have great teams that are going to pitch their ideas today, as well as 3 amazing judges that have so graciously offered to contribute their time and wisdom to judging this competition. So I want to introduce these judges now briefly. First, we have Lucretia Daniels, who is the CEO of Detroiters Working for Environmental Justice here in Detroit. We also have Mike McKay, and Mike is the executive director of GLEAR, or the Great Lakes Institute for Environmental Research. And last, and certainly not least, is Tom Zanicki. is the Agricultural Policy Director for MDARD. And MDARD here in the state is the Michigan Department of Agriculture for the state of Michigan. So at this point, we're going to officially get the party started. And so with that being said, I'm going to turn this back over to Felicia, who is going to walk us through our next steps.
Thank you, Paul. We're going to go ahead and get started straight into pitches, starting with ditch pollution. And so whenever you're ready, you should be able to share your screen, and I'll be able to start the timer.
Share screen. There we go. Okay, I'm ready.
All right, whenever you're ready.
Hello, my name is Chad Gaynor. This is my third Erie Hack, and I'm excited to present my idea, ditch pollution. The main issue of surface water quality is nutrient loading, and the largest offending tributaries for this are the Maumee and Sandusky rivers, so let's focus there. The main source of nutrient loading is fertilizer being flushed from fields into ditches during rain events, which moves down the watershed into lakes, creating toxic algae blooms and other issues. This is a map Central Michigan University put together showing the density of farmland in the area. And I started superimposing the watershed on top of it. But as you can see, the two rivers are surrounded by dense farmland. So coming into this event, I thought the solution was riparian zones, buffer strips that go between fields and ditches that would filter out any sort of excess nutrients in the runoff. But that's a big, complicated, expensive process that requires a lot of education and buy-in from farmers because of the land loss. This map gives an idea of the associated land loss and the complications with installing buffer strips. Another good idea is two-stage ditches, which it'd be awesome if we could just combine them, have buffer strips and two-stage ditches between farms and the watershed, but it's twice as complicated as just buffer strips. So I think we can achieve a lot of the benefits of these two solutions without any of the complications. This idea is fast, easy, cheap, and the only stakeholder involved in the implementation would be the counties that are responsible for ditch management. So we already have ditches around the perimeter of farms that are built to move water away from fields, but what if we sequestered some of the polluted runoff water in those ditches? This would only require a small amount of work and materials compared to the development of buffer strips and two-stage ditches, and it wouldn't create any land loss for farmers. Basically, we'd install a small metal levee in each block of farmland. Then when it rains, the runoff would pool along the farm that created it. And while sequestered, plant life would feed on the nutrients and some of it would settle into the sediment, but none of it would flow directly downstream except during very heavy rain events. But even then it would be diluted. So this is what I'm thinking. This is the current design. Really simple to install. Just weld it into the culvert in a ditch with one bead, and that's it. Put one per block of farmland. This would make ditches function more like wetlands, filtering pollution that comes through them. They'd also work more like sediment traps, which is a practice farmers employ to reclaim lost nutrients. Counties are responsible for ditch maintenance, and they typically place the dredge material back on the farmer's land so it can be reapplied to fields, which would continue. The only difference being the material would be far more nutrient rich than before. On the business side of things, I'm working on the design to streamline production. I have a CNC that can mill material up to 33 inches wide. I'm iterating a tool path to reduce material waste. I'm looking into material costs, and here's what I have so far. A 1/4 inch might be overkill, but I'm still figuring that out. Conservatively estimating 4 per sheet would put the material cost around $31 a levy plus production costs and markup. And I'm building a simple website where bulk orders could be placed. I haven't calculated the potential market yet, but if this reduces nutrient load in the lakes, we should install a ton of them. If we can install some of these levees in a test location, existing downstream sensors could monitor the impact of the solution. The red dots on this map are the sensors I was looking at. Adding a couple wet labs to monitor nutrient load before and after the levees would be ideal. And I'm also developing a dashboard using gldw.org data that focuses specifically on this region. Unrelated, but some potential additional benefits may exist in algae cultivation for biofuel, since it's pretty easy to run a tractor down a long straight line. And this is just kind of a theory, but changing the ecology of ditches should make them better hunting grounds for wildlife, which could reduce roadkill. So that's the idea. My next steps are to continue working on the levee design and manufacturing plan. I'd like to try this in the real world somewhere. I need to put more work into the sensor dashboard. I want to run this by someone at a county that does ditch management. And I'm also tinkering with some things for the water sensor mini challenge. Nutrient loading from farms is the problem, and it seems like sequestering runoff water in ditches could be part of the solution. So let's ditch pollution. Thanks.
Thank you for that presentation, Chad. Starting from this point forward, if all speakers could also have their cameras on as well when they present. But if the judges could, no worries. If the judges could now turn on their cameras and feel free to start asking questions.
My bad.
Chad, Mike McKay, Great Lakes Institute for Environmental Research, University of Windsor. I like the idea. A little daunting, just thinking about the Maumee River watershed. I think I saw a map where they looked at the drainage in the watershed and referred to it as a phosphorus superhighway. There's about 40,000 miles of drainage ditches throughout the watershed. So it's a massive undertaking, but I think you're spot on in terms of identifying, source of degraded water quality in the lake and to reduce nutrient loading. So ideas that can work towards that goal, I think really should be explored. From my perspective, scale is certainly going to be a big challenge. And the other thing, again, having lived in that area for some time, certainly we're in the spring. These ditches are there because that land used to be a swamp, and they drain the land effectively. So even when there are no barriers that might impede flow, during the spring, those ditches are sometimes overflowing. And, so you'd have to deal with, you have to sort of, run some calculations in terms of precipitation and expectations in terms of the capacity of those ditches to hold water. And do we have excess capacity where we could continue to hold water for longer periods of time?
Awesome. Thank you for the feedback. I have a lot of good points in there.
Hey, I have a couple questions, thoughts on this, slightly piggybacking off of some of Mike's. I agree. I mean, you definitely identified one of the bigger sources for surface water nutrient issues in the basin, and agree that there is a need to do more around water retention and detention for a whole host of reasons, both volume control, but also from a nutrient standpoint. For clarification, so is the idea, I wasn't clear in the schematic, is the idea that you would put that levee in a culvert, like in a road culvert or something, or?
Yes.
And then, so you would like just back it up in between essentially like two road culverts or two existing culverts? Because one of the issues that I'm not sure how you would get around is a lot of those ditches don't necessarily have a culvert running the whole way. So just curious kind of what that design is.
Well, I kind of thought we'd have to take a look at like the watershed map and figure out a strategic way to approach it. So some flows are going in the appropriate direction, and if you pick a good spot for a dam where the water would pool up in front of the farm, just do it where it's appropriate, because obviously everything isn't uniform. You have to look at each site and figure out what the appropriate location is. But the general idea is just pool the runoff directly in front of the field that produced it, and see if that extends any benefits.
Yeah, and so just in way of follow up, and as you kind of continue to further refine this, I think important to take a look at, and I'm glad to hear that you want to talk to a drain commissioner or water resources commissioner about it, but understanding what is feasible from like a legal standpoint and even from a political standpoint within the drain commissioner world, because backing up water in one place with a culvert, the landowners would have to pay for that, because those are petitioned projects that drain commissioners have to go to ratepayers to pay for. And so I think it might be a tough sell to tell landowners that you were going to back up water that can't then flow through the ditch for like the purposes of nutrient reduction, because that's not within the drain code. Nutrient and water quality is not one of the goals. It's all about moving volume off the fields and off of property as quickly as possible. So I think good to talk with some folks and just kind of figure out politically and legally kind of the viability around some of that.
Awesome. Yeah, I agree 100%. Thank you.
Thank you. I think that is all we have time for questions. Thank you, Chad, for sharing your idea. Next up, we have, if you can stop sharing your screen. Next up, we have Great Lakes environmental graphics. And if judges can now turn off their cameras.
Hello, can you hear me, Felicia?
Yes. Yeah.
I guess everyone else. All right, I'll start the presentation in a second. Okay, can you wait? No, I didn't. I don't think I shared my screen, did I?
No.
All right. Go ahead. Just full screen. How about now? Can you see me? Mm-hmm. Okay. So the team name, group name is Great Lakes Environmental Graphics.
Your time started.
Oh, right. So Great Lakes Environmental Graphics, our general goal is just to create a more accessible source of water and environmental news and information around the Great Lakes. This challenge statement is community water resources. So just briefly, my background, I'm an environmental engineer, used to work for the Detroit Water and Sewage Department and for environmental chemistry lab. And I've been a bit of an amateur graphic artist, made some t-shirts, book posters, sort of thing. So the main problem that I see in my daily life, but also in my professional life, was a sort of lack of accessible media about Great Lakes issues. When I worked for the city of Detroit, it was more about, infrastructure awareness. A lot of people didn't appreciate how old our combined sewer system is, why our combined sewer system's there, sort of a lot of the things that go behind where we are right now and why we're there. A lot of the media that I would see that is available to the public, it often contained too much text, didn't often give a concise background on the issues. And, more than anything else, I don't think the media really reached the public. And if it did, it didn't engage with the public. So I just pulled a couple examples from some of the, issue statements that we had with surface water quality. In general, these are good informational websites. They provide good information for people like us who care about these issues. But for someone who doesn't necessarily care about these issues right now, oftentimes it's loaded with text and information, and it's hard to read, and it doesn't necessarily engage them. And to me, I like infographics like the one on the bottom right, but oftentimes infographics, they're very information heavy, and for just a sole piece of information, it can be hard to sort of engage with the person without having a presenter or someone speaking behind them. My sort of idea or solution to that is I'm looking to make sort of graphic first media, utilizes a mixture of photo illustrations and data visualizations. And generally the text that is within it only serves as support. So it's sort of a focus on engaging and energetic presentation, a bit more fun, something to, keep the public interested. And so the media I'm creating is dual format. The primary format is a short form zine. I don't know if you're familiar with the zine. It's basically just a sort of independent magazine, sort of became popular, maybe not popular in the 60s and 70s. And it's really just you make some images, make your own zine and print it on your own computer paper, or you write it out and draw it and scan it. And so it benefits from very low print cost. And, you can go to you can go to your corner store or your gas station and you can distribute them because they only cost 30 cents to print. And along with that, there would also be a more web-based version for digital consumption. So I have a rough draft, very rough draft. It's more, just getting out the idea of, what it would be. And so Great Lakes Environmental Zine, sort of engaging picture. The first issue I was developing sort of centered around invasive species of the Great Lakes and sort of our history of, fortunately no longer, but our rivers burning. So, slightly more engaging cover, these invasive species being brought down. These would be pictures of the invasive species. And a focus on images that hopefully tell the story, and you can read text to get a background on them. So 19th century, what Cleveland or any industrial city in the Midwest looked like. 1868, our first fire happened on the Cuyahoga River, and then you can see as we approach the 1950s, they went up quite quickly. In the 1952, there's reports of 2-inch thick oil slicks. In 1952, the big fire, not the one that got people engaged in 1969, but the one that had all its images distributed in 1969. And other things like invasive species in the Great Lakes have, incredible potential for presenting the story in images. You can show elevations in the Great Lakes, that previously the Niagara Falls served as a barrier. We were basically an island with canals and locks. That all changed.
If you could start wrapping up.
Oh, okay, sorry, I'm almost done anyways. But telling these stories is lots of graphic potential. Anyways, so targets to develop the first first draft by November, complete the first edition by December, January. And then it's a pretty small goal, but any further work would be dependent upon whether actually it engages the audience. So I appreciate all your attention and time, and I'm open for questions and comments.
Awesome, thank you. If judges could now have their video on and ask away.
Thank you so much. I really appreciate this as a public health professional who is always trying to figure out how to engage laypersons and the type of work that we find important. And that seems to be what you were pointing to. I am wondering about how then you decide about language, or what sources you use to figure out sort of reading level types of language to use if you're trying to reach more lay people versus those of us that are typically engaged in this work.
That is something that do you mean like what specific sources of information text I would reference, or what sort of standards I would follow?
Both.
And so I haven't found a good reference for, this is exactly how you present the information to the public. And it's something that sort of challenges me anytime I make any sort of graphic media, book in the past posters, is how to put the information to the public and have them read it and understand it. I don't know if I have a good answer other than it requires, revision, drafting, and having the public read it to make sure that they're getting what they need from it. And then, redeveloping.
That makes sense. Just to point you to a resource, so the CDC has a guide for plain language.
Okay.
So that might be something that would be helpful, because a lot of these concepts that we deal with are really complex, really big words. And so that might be a good guide for you. And also, I would just encourage you, as you're developing the zine, to think about, besides accessible language, translation as well. So thinking about the regions where you would be disseminating it, and whether translation is necessary and in what languages.
True. That's a good point. Thank you.
Robert, I agree wholeheartedly with Lucretia's comments about science communication and increasing awareness amongst the public, and we always struggle as to what's the most effective way of doing so. So I appreciate your efforts in trying to do this. I'm wondering if actually your zine images of the Cuyahoga River burning brought to mind the marketing campaign of the Great Lakes Brewing Company in Cleveland and their Burning River Pale Ale. And I'm wondering if if working with private sector and having messaging on commercial products can also be an effective way of communicating increasing environmental awareness. Because again, you see magazines at a gas station or something, and I guess some people grab them and read them, and some people don't touch them, and some people pick them up because they need to wipe a spill off or something. So just again trying to think about the most effective way to reach people.
Yeah, and I think when I said gas stations, my original, I think a lot of opportunity is to work with private industries like the Great Lakes Brewing. They have that, the beer, but also, places where, people aren't just stopping by, but where people spend a minute of time. I live in East Detroit. And so when I think of gas station, I also think, most gas stations have a pizza or chicken. And I was thinking places where people have to stand and wait and be there for a minute. But also, working with industry, that's a good idea.
All right. Well, thank you, Robert, and thank you, judges. Our next pitch isn't until 3:40. So we're going to take a quick 5-minute break and get right back to it. So feel free to have your cameras off, and I will see y'all later. All right, welcome back, everybody. We're just gonna give the judges another minute to get their things together. But yes, thank you, Amit, for sharing your screen. All right. Well, whenever you're ready to start, I will go ahead and start the timer. So whenever you're ready.
Thank you, Felicia. Dear judges and audience members, thank you for this opportunity to present. My name is Amit, and my team member April and I will be presenting our project, Aruna Innovation. Aruna is founded on the principles of people, planet, and prosperity, in joining the mission of preserving our finite and precious resource, water. The problem is large and needs to be addressed. Lake Erie and similar water bodies are being affected with several contaminants such as heavy metals, PFAS, pesticides, all kinds of microbiological contaminants and algae blooms. Lake Erie is a great lake, and let's keep it that way. Now, due to these challenges, we need innovation in the water treatment space, a treatment method that is faster, safer, and scalable, and more importantly, environmentally friendly. So we've come up with a solution called Triton, which is based on the premise of pulsed UV. It is a novel water treatment method, and in early 2020, we started to perform some tests with unfiltered water from the Hudson River. As you can see on the screen, the before and after pictures, just after a few seconds of treatment time, within 24 hours, we had zero regrowth, whereas the untreated water, you can't drink it. And it's pretty evident with the pictures. The value proposition is based on months of R&D and several validated studies. Pulsed UV has the following value proposition. It has lower operating costs. It's FDA approved as a technology. It eliminates all bacteria and viruses. And we believe it can also degrade and eliminate pesticides, PFAS, and chemical contaminants. And like I said, more importantly, it is environmentally friendly, which means it does not use any chemicals or mercury during the treatment process. The picture that you see is a picture of our benchtop unit, and we are in the process of building our scaled up unit. This picture that you see is our commercial pilot that can treat up to 60 gallons per minute. We are very close to finishing it, and we can deploy it as a pilot in the field to test on different kinds of contaminated water and get results and keep moving on. Now, when we are ready to commercialize, we did a quick scan of the market on what the market opportunities are. And we came up with some of these lower hanging fruits that require immediate attention: industrial wastewater treatment, aquaculture, and agriculture. Now, this graph is based on the stringent needs in the market and the quality of water, the purity of water based on the market need. So we believe industrial wastewater and agriculture, aquaculture could be a really good target as and when we are ready to get to commercialize. My team, this is myself, I'm the founder and the founder of the technology. I have Shekhar, who is the co-founder, and he's in charge of strategy. April is our marketing associate. Jennifer and Samir, they bring a wealth of knowledge in water contamination. They are both in the water industry. Samir is a PhD in environmental engineering, and Jennifer runs a lab at one of the water treatment facilities in New Jersey. The foundation of Aruna, again, is built on people, planet, and prosperity. And we believe we have strong alignment with the mission of the Erie Hack Challenge and to protect and preserve Lake Erie for the great lake that it is. So please help us in joining this. Thank you very much.
Thank you, Amit and April. If the judges could now turn on their cameras and ask away.
Yeah. Hey, this is Tom. interesting presentation. I appreciate the time. I have a couple questions for clarification. So first and foremost, what is the pathway that the treatment takes? Like, I understand that you probably can't share or don't want to share the exact mechanism that the treatment occurs through. But admittedly, without a little bit more detail on what the mechanism is, I'm skeptical.
Sure.
And then kind of a follow-up question, and maybe you can incorporate it into that answer, is the regrowth slide that you showed. To me, that lends more towards like the biological piece of it, and the bacterial side of reduction. But to do everything from bacteria to viruses to PFAS, there's a lot there. So can you speak to all of that a little bit?
Absolutely. So pulsed UV is not new. It's been around since 1970s. It's light-based technology. It's broad spectrum light as compared to our mercury UV, which is extremely narrow band. So when you look at an effluent, the water goes in through a 30 and a 50 micron filter with our pilot unit, and it removes any kind of visible contaminants. And the stuff that's not visible, the stuff that's deadly, is treated through our pulsed UV unit. The second part of the question is about microbiological contaminants versus PFAS and chemicals. So there are two things to this. When you expose UVC rays to any organisms, they're destroyed based on the dose. So that's one thing that we do at a very, very high level, because we pulse it with a lot of intensity. Same thing with chemicals. And there are several studies, for example, NDMA, which is a byproduct of rocket fuel. They were able to take raw water from the Colorado River, which had concentrations of NDMA, and reduce it to 99% with one pass. So when I mentioned that we've done a lot of R&D with studies, these are some of the studies that we've proven, and we've tailored the unit based on the amount of energy required in order to not only eliminate microbiological contaminants, but also chemical contaminants that break the bond.
Yeah, that's appreciate that. I admittedly may have missed that the entire process was through pulse UV. But the additional explanation is helpful. I don't want to monopolize all the time because I think Mike might have a question too, but I'm hoping at some point you can also talk about cost and the energy intensive piece of it, or like what the energy needs are to run the system.
I can give the cost we haven't gotten there yet because we are still in the pilot phase, but from an energy perspective, it's up to 70% less energy compared to traditional UV systems when you look at gallon for gallon treatment.
Great, thanks.
Awesome, thank you. I think we have time for one more question, and then I want to make sure to give time to the judges to fill out their scores. So if Mike, you have a question.
Thank you, Felicia. Yeah, thank you. Nice presentation, and I'm a little familiar with the use of UV technologies. We've got a company in Canada, Trojan Technologies, that uses UV often with wastewater treatment plants. Now you're marketing this for towards it could be universal, but this is a Great Lakes competition. Thinking about some of the challenges we have in the Great Lakes in terms of water quality, I'm thinking, drinking water, for example. So, we're all familiar with problems we've seen in Flint and on the west coast of Michigan, Benton Harbor, I believe, with lead in lines. Any influence of pulse UV on metals? I know you mentioned metals earlier in contaminants.
I have not yet. So the heavy metals is one aspect we haven't tested it on. And I would love to have the pilot scale being tested on heavy metals. But the premise of pulsed UV is bond dissociation. So any chemical or contaminant that has a bond, it will destroy the bond and degrade it. So the heavy metals being a single ion, I haven't read any literature that does that or any studies that have done that, but we would love to try running lead contaminated water through our unit and see what happens. And that's the whole reason we have a bench scale unit, which has never been done before. So we are the first to scale it up from a lab to 60 gallons per minute.
Thank you.
Thank you.
All right.
Great questions. Thank you.
LaPrisha, do you have a question? Okay, thank you. All right, I'll just give it till 53 for y'all to fill out the form, and then we'll get started with our next team if we can go ahead and get everyone situated. But thank you all.
Thank you guys very much for your time. I appreciate this.
Thank you.
I'm also looking for my timer, which I think may have disappeared. If judges, are y'all ready? If can y'all give me a thumbs up if y'all are ready? And we may have to just, I may just have to give y'all signals through the chat.
Do I have to share the presentation, or we emailed it to you? So are you putting it up, or am I putting it up?
If you're putting it, you emailed it to me for backup, but if you have it now, you can go ahead and put it up.
Okay.
I can also put it up, whichever one's easiest.
I can do it. That's fine. Looks good.
Okay. Whenever y'all are ready.
Okay. So I'm Dr. Kevin Hagedorn. I'm an optical engineer. Our company is Life Magnetics. So we were funded by the National Science Foundation to solve a very specific problem with RNA shipping and storage. And eventually we'll get to how that relates to wastewater. So do you remember how we couldn't do at-home COVID-19 testing and the vaccines needed to be stored at -80? Well, it's a very common problem in molecular biology and RNA testing in particular, that RNA is not a very stable molecule. It degrades relatively easily. RNase, which is an enzyme which degrades RNA, is one of the most ancient and conserved mechanisms in biology. So we were funded by the National Science Foundation to use a quantum mechanical mechanism called pi-pi stacking in order to address this issue. And I'm not going to talk about what any of that means. However, what the technology does and how it relates specifically to wastewater testing is that when linear single-stranded nucleic acids are present in a solution with calcium chloride, the carbon will bind the RNA in solution. So this is the carbon beads distributed in solution. And if there's RNA present, then you get this binding and it makes these clumps. What we've done with this technology as an initial application is apply it to population screening for COVID-19 in wastewater. So we did this in collaboration with CMU. This is actually all data from Central Michigan University. The old technique that they used to use for getting this data was based on precipitating all of the solids in the wastewater sample. So over on the left-hand side, that's effectively what all those steps represent, is they take the raw wastewater sample and they go through a number of steps that take 3 or 4 days to basically precipitate down any solids which are present in a liquid, and then they run through standard extraction methods. Overall, the process takes about 3 days. However, we've been able to show that with a quick 30-minute extraction, we can directly pull RNA out of the liquid with calcium chloride and a little bit of soap. And this allows us to do population screening for COVID-19 in wastewater, even though this is a very low abundance target. You imagine somebody sneezes and it goes down the drain. There's then not very much COVID in that wastewater sample. However, using digital droplet PCR, we can actually quantify all the different variants of COVID-19 in a single water sample. So the way that this works, this is a digital droplet PCR. So you break up the sample into millions of tiny droplets, and then you amplify, you reverse transcribe and amplify the genes that you're looking for in each of the droplet samples, and then statistically analyze them. And based on the base mismatches, we can tell which variants are present in a sample. So I'm not a biologist. However, we did some background research to make sure we weren't totally off base. So from a 2005 study, the National Institutes of Health concluded that in many cases, microscopy was insufficient to tell the difference between toxic and non-toxic populations of cyanobacteria. In these cases, they had to use PCR to determine which algae were toxic and which were not. And going on a little bit further, so every cell in your body has the same DNA. So every cell in your body is capable of producing, for example, gastric acid. Now your eye cells don't, and if they did, that would be bad. So there's a difference between having the capability to do something and actually doing it. And this is where RNA expression analysis comes in. With RNA expression analysis, you can actually tell whether or not something is a molecular mechanism is being performed or not. And so if we were to apply the wastewater testing technology to cyanobacteria populations, we should be able to tell, one, whether they have the capability of producing the toxins, and two, whether they're actually doing it. And given that we can detect COVID-19 in these water samples, probably the algae, there's more than enough to do RNA-based detection. So what we're proposing to do is seeing how little work we can get away with to do the accurate detection of these toxins. In the simplest case, for example, if we don't need to break open any cells, it should just be a matter of adding calcium chloride to the water, pulling out RNA from the sort of dead or already lysed cells, and then doing digital droplet PCR. We can also see if we can get away with a more portable technology like LAMP. If we need to digest the cells in some way, like with sonication, that can make it slightly more complicated, but it still should be fairly reasonable. Worst case scenario would be having to use some sort of chemical method like Trizol to lyse the cell populations. And the target would be, at maximum, this shouldn't be any more expensive than a standard COVID test. So it's $50 per test for 96 at a time, or $200 for a single test. But I imagine it would actually be cheaper than that, but I haven't specifically calculated how much it would be. And the second part of that, so RNA is extremely.
I'm gonna call time now.
Okay.
Sorry about that. Judges, if you could turn on your cameras and start asking away.
All right, I'll kick it off because and this is it's I appreciate the presentation. Obviously, like the toxicity issue around algal blooms is a huge concern, and I think what really drives like the need for a better health-based response to some of this to nutrient pollution issues. I'm curious on how much you have ground truth this with microbiologists and with folks that study algae populations really intensely, just because I thought a lot of it is driven, well, they don't really know of what turns a bloom toxic or not, and if it's a differentiation between N and P balances within the water or what the algae has access to. So I'm just curious kind of what level you guys have done to verify this.
Yeah, so actually, Kayla did a little bit more work yesterday than I did on differentiating. It's a complex issue, right? And as far as we can tell, we would need to verify with a molecular biologist if they think there are specific genes associated with expressing these toxins that we could use for as an indication. We know the 2005 study found the genes in DNA sequences, so they could tell if a population was capable of producing the toxins, but that wouldn't necessarily say that they were. And I think Kayla found some literature yesterday indicating that it's actually a symbiotic relationship, which, go ahead.
Hi, I'm Kayla. I'm a scientist that works for Kevin at Life Magnetics. And like you said, I was doing some research yesterday on literature. And from what I found, there is a hypothesis of a symbiotic relationship between the dinoflagellates and cyanobacteria. But that aside, there are two specific genes that researchers had found in the literature that I had read, SXT, A and G genes, that were solely in the toxic algal blooms. And so the biggest thing that I think we were talking about and thinking of is doing PCR on the algal blooms if somebody even has an inkling, like if they do a water test and there's high phosphorus or nitrogen and they just want to test the algae, we could potentially detect before it becomes an HAB. And they could treat the water and the algae at a really early stage instead of allowing it to get to a point where then it's causing a problem to aquatic life, human life, as well as, you know, the whole biome. And my background is biology and microbiology.
So I imagine there could be three sort of situations. One, you do the algae test and it turns out the algae are not capable of producing the toxin. And we determine that through PCR testing, DNA testing. The other situation would be the genes are present, so they're capable of producing the toxins, but they are not currently producing the toxins. There's no RNA expression of those particular genes. The third situation would be that they are actively expressing the genes to produce the toxin. So you could differentiate between those three sort of states in the sample.
I think we have time for one more question.
Yeah, just so I guess right now the state of Ohio approves a qPCR based assay for looking at potential toxin production. There's two approaches. You can either measure the actual toxin in water using an ELISA assay or something, or MS, LS. LCMS or the qPCR assay, but you're right. That just looks like that potential. It doesn't actually measure expression. Yeah, I mean, people do look at the expression in water. Your comment.
Sorry, if I can just add on that slightly. So, I mean, the major innovation that we did at Central Michigan University before our technology, it took them 4 or 5 days in order to purify the RNA out of a sample in order to actually do the COVID population screening. So if we made it easy enough for the lab that you're already working with to do RT-qPCR, I would be happy to work with them for them to actually run the qPCR assays. We would simply give them a tool for them to better be able to purify the RNA out of the water in order to run it to begin with.
Yeah, no, I agree. Having that that initial step of collecting the sample and concentrating and extracting can take time. I mean, one of the issues right now, if I look at the drinking water utilities, for example, who are mandated to do this testing, oftentimes they're taking the sample, then they're shipping it off to a commercial lab, and so you're already looking at a few days. So having something that they can do in-house, and you talked about maybe expanding this to use LAMP or something like this, something in-house that provides a rapid answer, has value to it.
So I can add on a little bit more for what we're doing with CMUs. So our long-term goal with them is actually to basically purify out the RNA on site and not ship the water, but ship the RNA bound to the beads like you saw in that one picture, and that would simplify the shipping and storage. Now, that we didn't do that as the first step because it has to change their workflow, right? It changes their workflow, and now the people on site have to collect those little vials, and they need to be trained to, how to get the RNA onto the magnetic beads and like that sort of stuff. So the simplest first step was shipping in the water and then just using this as an extraction of the water sample that they get into the lab. But ultimately, what we would like to do is basically just ship in 1.5 ml vials of those little beads with the RNA already attached that you collect in the field to limit your shipping and that sort of stuff. And that also, since the RNA is continuously degrading, you have RNase activity in the water sample. So the RNA is continuously degrading as you're shipping it. If you don't do something like attach it to the magnetic beads for storage. So it's better if we can stabilize the sample on site, but in the meantime, we can still make improvements.
Oh, great, thanks.
Awesome, thank you. Okay, so next up we have Andrew from Stark Drones, but we're gonna start at 4:10 just to give the judges enough time to put their notes together.
Okay, can you guys hear me or?
Yes,
No?
just 1 second. Judges, are we ready?
I am, yes.
Okay.
Yes.
Okay, I will start right now. I will be presenting underwater wireless sensory networks. In the past, I've built numerous variations of underwater wireless sensory networks in order to collect real-time data about oceanic contamination. This time, I propose another real-time sensory network that utilizes various upgraded telemetry techniques, such as an extra data syncing peer in order to improve the data collection process. The main problem, contamination in the water can cause some serious danger. There are different variations of flesh-eating bacteria, parasites, harmful algae blooms, and things like that. Timing and the collection of data is key. Every minute counts, and things such as chemical or wet testing methods take about 24 to 48 hours for this species alone. And also, it's a productivity problem in regards to trying to get real-time and faster data. Some testing methods cost over 30K in regards to wet and chemical testing. A lot of this is put through our taxpayer dollars, and over 485,000 people a year die alone from lack of clean drinking water, especially in third world countries. We want to dramatically lower that. A lot of our ocean is also unexplored, and 70% of the planet's surface is covered by the ocean. So obviously we want to preserve our ocean. So previous solutions in regards to tackling the problem, I've built variations of different sensors that were $125 to $150 in order to collect real-time data. Part of it is not just the development of the sensor, but also the wireless telemetry method that I created. The first solution I've done for a prototype had problems with enclosure design and deployment, but the second solution fixed that, and upgrades in regards to method for signal processing was done through an optimized sonar converter. We ended up creating a wireless network that actually allows for higher data extraction efficiency, and tests so far have been basic lake deployment in regards to previous solutions. Lab environment testing is available with access to higher resources and or funding. These are the variations of the different prototype phases. The one on the corner is obviously like the bad enclosure one. I also have my own telemetry module that has a LoRaWAN module built in it, as well as a transceiver module and custom software code. This small device right here, which is rechargeable, replaces a telemetry module that's almost the size of a fridge for a software-defined networking. And I have a basic prototype right here, which I could go over more about the prototypes and R&D in the Q&A. This technology has been presented in front of the Department of Energy Environment and Gigabit DCX, MakerVair. I talked about this in some of the Internet of H 2.0 competition, and I also presented this, gave the same presentation recently in Lakebed 2030, as well as in front of top scientists from NASA. In regards to the current solution, we created our own mesh and data syncing protocol for telemetry. And we also created different things in regards to different power-up methods, different grid implementations, and different algorithms that are able to work with different sensory designs that we have. In regards to the hardware, we've built our own development board in regards to the telemetry module. So this would allow us to end up manufacturing thousands of sensors. And we also created variations of optical mass spectrometry sensors and sensors that are really cost efficient compared to the competition, as well as various different data syncing modules. Our goal is to eventually have tens of thousands of sensors deployed, targeting mineral offloading and cleanup for various parts of the ocean, and basically building Internet of Things for the ocean. I think we can do that given that our IP, most of our IP is protected, and the PCB designs we have are extremely scalable for additive manufacturing. And these are the sources. Now I welcome different Q&A.
Thank you, Andrew, for sharing. If judges can now turn on their cameras and start asking questions.
Yeah, a clarifying question. Appreciate the presentation. A lot of good information and concepts there. Could you speak a little bit to how you would specifically deploy these in the Great Lakes and in Lake Erie specifically, and then how this technology or these sensors differ or augment existing technology that's already out there?
Hi, yes. In regards to deploying, we have our own sensory design. Our sensors are like extremely portable. So with funding, we could obviously manufacture like tens of thousands of these. Right now, just because of lack of access of funding, the current design is based off of, you could just unseal the enclosure, recharge it, and then put it back in. With funding, we'd obviously be able to do things with hydroelectric and wireless grid. In regards to things like these are extremely portable. So you could build base stations with lots of these modules, as opposed to something like this. I don't know if you could see it that well, but there are telemetry routing devices in regards to the current technology that are extremely expensive, about the size of a refrigerator, hard to move. With our sensory technology, it's obviously way more portable, and with portability as well as cost efficiency, you're able to deploy much more. And lowering the cost in places beyond Lake Erie, like different places that we were unable to explore before, now the cost is so much dramatically cheaper that if you wanted to do mineral targeting or targeting different mineral offloading places for like harmful algae blooms, nitrate, phosphorus, viral bacteria, you could end up monitoring and doing automated targeting for those locations. So.
Andrew, do you envision, you're talking about creating hundreds, if not thousands of these to deploy in the lakes. Are you envisioning these to be stationary? You know, when we think right now of sensors in the Great Lakes, they're often attached to buoys, and they are stationary via usually a pyramid anchor or something. Or are you thinking about just free floating, like a drifting type of approach?
In the beginning, they would have to be stationary. In the future, obviously, when things are more automated, a lot comes with funding. We do a lot in regards to nanotechnology and different designs for optical spectrometry. So in the future, you could actually have robots deploying these sensories, and they could be free floating. Currently, right now, it'd be buoy based, just on the lack of funding. I mean, we already have designs for free flowing ones as well as stationary ones.
Okay, thank you.
Are there any other questions for Andrew? Okay, well, thank you so much, Andrew, for presenting. So what's going to happen next is the judges are going to turn off their videos and mute themselves, and we're going to go to another room where they're going to deliberate, and we will eventually come back to announce who is moving on to semifinals. So feel free to stick around for that. In the meantime, you can enjoy this beautiful video that was made by our partners. Michelle?
Hey, Felicia.
Yes.
Is the other room Google Hangout?
Yes, it is on our calendars, I believe.
Yeah, I'm not going to be able to access it because of the state computer doesn't like link up to Google stuff. So, I guess there should be a call in though with it, correct? Just like a phone number.
Yes, there is a phone number.
Okay, I'll just call that. We're good.
Okay, cool. I'll see you there. Hi, so the judges and are making their way back, but and but in the meantime, um, so we're actually still waiting on Paul because Paul will be back to announce the winners and who is moving on to quarterfinals momentarily. Speaking of which, just in time, as always. So Paul, take it away. You're on mute, Paul.
Yep, I'm off now. It's perfect timing. I just got back into the room. So as I'm sure Felicia extended a huge and warm thanks to everyone who participated today. As I promised, it was going to be a lot of fun. I strongly encourage whether you move on to semifinals or not, I think you still continue to pursue these innovations and ideas and continue to leverage the ecosystem here and across the Great Lakes in order to try to move these ideas forward. I do want to say that we have two winners today and two winners that will move on to the November 4th regional semifinals. Those two winners are, drum roll, Life Magnetics and Stark Drones, both of which are winners of $2,500 each that will hopefully make an impact and contribute to the evolution of the innovation that you guys have presented today. You can contact me. I think you guys both have my email address, so you can contact me offline and we can make sure that we take care of those logistics. But also, I strongly recommend, like I said, not just the two winners today, but also all 5 presenting teams to contact our judges and to contact me, because we have a wealth of resources across our ecosystem in order to provide feedback and guidance and just thought partnership in how you proceed as you move along your entrepreneurial journey. So congratulations. And I didn't mention this in the beginning, but I do want to give a personal and explicit and direct thanks to TechTown Detroit and the entire team. Michelle is on the line today and really helped to ensure that this moved through as seamlessly as it did. So I definitely want to thank TechTown Detroit, WEtech Alliance, Wayne State University as core partners here in the Southeastern Michigan and Southwestern Ontario region. So thanks again to everyone and congratulations to our winners today. And now, Felicia, I'll turn it back over to you.
Thank you, Paul. And thank you again to all teams that participated for your hard work. And congrats to Life Magnetics and Stark Drones. You will receive an email from me either tonight or tomorrow regarding next steps and for additional contact information. Thank you again. And I can't wait to see what's next.
And Felicia, 5 on the nose, perfect timing.
Yes, we love being on time.
Great job.
All right.